Hello, I am Anup Singh from Sandia National Labs in Livermore, California. I'm a chemical engineer and I make small things for use in many diverse areas such as medical diagnostics, for lab research, and today I'm going to talk about SpinDx. It's a small device, as you can see in the picture on your right, and there is a CD that I'm holding in my hand that goes into that device to run these assays that I'm going to talk about. It's a platform which can run as many as 64 assays in a matter of minutes using a very tiny amount of blood, saliva, urine, or other samples. Why are we so excited about it? Because it's faster, better, and cheaper than what exists right now. It is very inexpensive, costs less than $1,000 for the reader and less than $2 for per assay or the disk. It has analytical sensitivity, which is better than most of the assays that are available on the market right now. It can run 64 assays at the same time, and it's very fast. It can do all 64 assays in less than 15 minutes without requiring any sample preparation. It's a plug-and-play device which has two components, the reader, which you see in the figure to your right, and a CD or a disk that contains the disposables to run the assay. All a user needs to do is insert 2 to 20 microliter of whole blood. That's the volume of a pinprick blood into the disk, load the disk into the reader, and then hit the start button to run the device. That's how simple this is. It takes about 15 minutes to run all 64 assays, and then the results are displayed either on the LCD screen of the device itself, or you can have a computer connected to the device that can take the data from the device. The disk is single use, but can run as many as 64 assays at the same time. SpinDx is based on a novel sedimentation technique, which combines two very novel approaches. First one is bead-based assays. So the CD contains beads that have the reagents attached to it, and these beads are mixed with the sample. Then the CD is spun, and then the beads are read using a fluorescence detector. How is it different from many other centrifugal devices that are on market, as shown in this picture from Samsung, Abaxis, and Gyros? Our method is very different. No other platform uses the combination of bead-based assays together with the sedimentation principle to enable a very sensitive assay. The combination of high surface area offered by the beads, integration of signal over many beads being read at the same time, and complete removal of the original sample gives it sensitivity that is unsurpassed. Because of the sedimentation principle, you also do not require any sample preparation. You can load whole blood and hit go, and the device will run your assays. To point out the sample prep example, here is a picture where we show how everything in the sample other than your analyte is left on top of this density media. So you can have cells in whole blood, you can have other proteins, they're all contained in the top of this density media, while the beads that capture the analyte goes to the edge of the CD, and they are read by the detector. We can also run as many as 64 channels. So I showed you the picture of the CD. Think of a bicycle wheel with spokes. That's the concept we use to have multiple channels in the same disk. And you can have as many as 64 of these channels in one disk, and each channel can run a different assay, allowing you to do as many as 64 markers at the same time. The device is very small. It's about cube of 5 inches, and the disk is 4 inch. The disk is exactly the same size as your CD or a mini DVD. So wherever possible, we have tried to use standard dimensions to build this device and standard components to go into this device to keep the cost down. The sample to answer time is 15 minutes for most assays, but there might be examples where you need extreme sensitivity. And then in those cases, you may add some additional time and going for as long as 30 minutes. In our hands, the device costs less than $1,000 to build, and we expect the disk loaded with the reagent to cost less than $2. The device is at the technology readiness level of 5 to 6. We have a working prototype, and we have many different assays developed. Some examples include cytokines, markers of heart disease, immune response markers, biotoxins. In terms of commercial readiness, we think that the discs are very easily manufacturable using very inexpensive plastics. Literally, you need 2 pieces of plastic glued together with a double-sided tape to make the CD. That's how simple this is. The beads that are the disposable reagents in the device are commercially available, and you can buy them by grams for less than a few dollars. The device itself uses standard commercially available components. And again, the point was to keep it as inexpensive and as simple as possible for you to scale it up into larger productions. We have also been very good at filing patent applications. We have 6 US patent applications pending right now, and we already have 2 licenses, but we are hoping for a lot more for very different fields of use for this device, because we are really excited that this device can make a difference to a lot of very different areas that you may worry or care about. What we are looking for is partners to commercialize the technology. At SpinDx, while we are very good at developing assays and engineering these prototypes, we just don't do any manufacturing or any selling of the device. And that's what we are looking for people like you to come in, license the technology, work with us to take us to the next step where it gets out in the market to be used by other people. This exciting technology, developed by Sandia National Labs in Livermore, California, is ready for licensing. If you are interested, we are happy to engage and answer any questions you may have. Here is a slide which gives you information for who to contact. Thank you very much.